postdoc in Dr. Catherine Lennon's lab at the University of Kentucky, and I'm really excited to talk to you guys today about some work I've been doing testing predictions of the adaptive decoupling hypothesis using transcriptomes that I obtained from different life stages of a hypermetamorphic insect, Differential, which is this cute little guy blowing a little bubble at you. So first, it takes a long time to load. Okay, so I want to tell you a little bit about the adaptive decoupling hypothesis. So metamorphosis is at the base of the adaptive decoupling hypothesis, and as you can see, it is spread throughout the entire tree of life. And what's really interesting about metamorphic lineages is that they're really diverse. They're really good at diversifying, and among these, the holometabolous insects are the most speciose. And so in these lineages, we have larvae and adults that are separated by a pupal state. And so the larval body completely disintegrates into imaginal discs and then reforms as an adult that's reproductively active. And the adaptive decoupling hypothesis states that the reason that metamorphic species are so speciose is because metamorphosis reduces genetic correlations and enables the independent evolution of different traits being expressed at different life stages. So Haldane first proposed some of the logic behind this in 1932, and then Nancy Moran really fleshed it out in 1999. And it all has to do with sexual antagonism. But we can imagine a trait or a gene that is expressed in both the larvae and in the adult. If selection is acting in the same way in both stages, and let's say it's beneficial, then any new mutation that pops up is going to be allowed, and hopefully, if drift doesn't get rid of it, be fixed in the population. So evolution is permitted. But now let's imagine a case in which we have antagonism between the two life stages. So a mutation arises, it's good in the larvae, but really bad in the adults. Natural selection is going to wipe it out and not allow it to be fixed in the population, and evolution is ultimately constrained. The solution to this is to decouple different traits or different genes expressed in the different stages. So we have one gene or one trait being expressed in the larvae and a second one in the adult. And when a mutation appears that's good for the larvae, evolution can be permitted, and even furthermore, additional ones can arise in the adult and be enhancing this evolution. So I wanted to look at this in Differential, and I argue that this species is hypermetamorphic. And so what this means is that there's multiple metamorphoses in this life cycle. So here we have cryptic larvae and aposematic larvae, and we argue that there's a minor metamorphosis that occurs between these two stages. The cryptic larvae and the aposematic larvae are both gregarious, and they both are feeding on pine tissues. However, they have different behaviors, with the cryptic larvae being lightly colored and kind of blending into their pine needle and eating only the exterior of the needle and not eating the resinous core, while the aposematic larvae eat that resinous core and actually use it to defend themselves against predators by rearing back and blowing the spit bubble that their predators don't like. We then have an intermediate metamorphosis event between the aposematic larvae and the wandering larvae. And what's really interesting in this system is that there's an entire instar devoted to this wandering stage. And these wandering larvae are solitary, they're non-feeding, and they don't care about their buddies, they just want to find a place to spin their cocoon. And then, as with all holometabolous insects, we have this complete metamorphosis event between the wandering larvae and the adult male. Oh no. Okay, this might happen all throughout, I'm sorry, I apologize. Okay, so here you just see those three metamorphic transitions, and we expect there to be variation in the amount of antagonistic selection across these different stages. So I don't know if you guys have noticed a trend, but ecology seems to be a big factor in these life stages, right? And so here we have a very similar ecology versus a very different ecology. And we expect that antagonistic selection increases across these different metamorphic boundaries. And that's where the adaptive decoupling hypothesis comes in, with us having lots of coupled traits or coupled genes in these cryptic and aposematic stages versus lots of decoupled genes that kind of goes along with that antagonistic selection and the wandering adults the wandering larvae and the adults. So this leads to two primary predictions. The first is just that decoupling should increase with the degree of metamorphic transition, and this is really coupled with ecology. And then the second is that genes under antagonistic selection should be more decoupled. And one of my favorite data sets is transcriptomes. And so here I took the head and body tissues of these 4 different life stages, and looked at 9,304 genes that are expressed in these tissues. And what I want to also draw attention to here is that we only use male larvae, so that we don't have any confounding sex-specific factors. So I first tested this question of decoupling increasing across the different metamorphic transitions, and we did this in 3 ways. The first was to look at a principal component analysis and simply ask, do the different life stages cluster independently? So I'm showing body tissues in yellow and head tissues in gray. And you'll see that we had 4 biological replicates for each of these samples. So here we have 4 biological replicates of the cryptic larvae, and they're clustering right on top of those 4 from the aposomatic larvae, right? We don't have any real distinction between these 2 different stages in the bodies or in the heads. However, we do see an independent clustering of the wandering larvae, which does have that slightly more intense metamorphic transition, and this becomes even more extreme when looking at the adults. So here are those adults compared to the wandering larvae, and as you can see, there's a lot of kind of space between these. They're clustering pretty far apart. And so we definitely do see this, and we do see kind of this trend that we expect across the different life stages. We then looked at differential expression. So, okay, so the volcano is exploding. These are volcano plots, and my lines didn't quite match up here, but no expression, or no change in expression between the cryptic and aposomatic is going to be right around the 0 mark, and anything that's black is not significant, while anything that's in red is significant. And as you can see, there's a few genes that are significantly upregulated in the cryptic, and a few genes that are slightly upregulated in the aposomatic larvae, but there aren't that many, right? If we watch another volcano explode, you're going to see that there are going to be even more. And I'm just going to skip ahead and let it start loading the next one as well. So we're going to get progressively more and more genes being differentially expressed in these different life stages. And this is going to load at some point. Wi-Fi connection issues seem to be the name of the game at this meeting. Okay, so we're going to see this increase in decoupling as we progress across these different life stages. Watching all the volcanoes explode. Here we go. Okay, see that huge increase between the cryptic and the aposomatic versus the aposomatic and the wandering, and then this crazy increase. And this actually, the significance even goes higher, but I wanted you to actually be able to see the cryptic and the aposomatic comparison. And I'm going to kind of preload the next slides too. Oh no, they're going to be fine. Okay, cool. So the next question that we wanted to ask was whether or not the correlation of gene expression also decreases with dissimilarity. So here I'm showing you the log of the expression in the cryptic and the aposomatic larvae, and each of these dots represents a single gene. Any genes that falls on this one-to-one dotted line is perfectly correlated, so we can predict the expression of the cryptic, so the cryptic using just the aposomatic and vice versa. However, there are some genes that are kind of coming off, and a lot of those genes are the same ones that popped up on those volcano plots. And now it's going to take forever. And what I'm going to show you next is that that span away from that one-to-one correlation line is going to be increasing and increasing in the aposomatic versus the wandering tissues, and then again in the wandering versus the adults. And I skipped ahead a few slides so it could load. And again, you're going to see that coupled to decoupled gradient with an increase in decoupling as these stages become more extreme and as these metamorphic events become more extreme. So I hope these 3 comparisons have convinced you that, yeah, no matter how we look at this data set, we see that as these metamorphic transitions become more extreme, that we're seeing more extreme differences in the genes themselves. So then I wanted to test that idea that we have these different genes that are under antagonistic selection, right? So some genes are going to be under antagonistic selection, and then other genes should have similar selection regimes. And to do this, we're going to use the natural life history of Neogypian lecantii, and this life cycle is really well known. So we see, just by looking here, we see that we have these cryptic individuals that are a different color than the aposomatic, and wandering, and then the adults are a different color as well. So we might have genes that are involved in pigmentation that are going to be decoupled, while all of you guys will be having the same sets of genes that I would pick for your categories that are coupled, right? So things involved in cellular localization and cell cycle are going to be coupled, while genes involved in things like pigmentation are going to be decoupled. Other things that are going to potentially be decoupled are things involved in immune system responses. So these guys are exposed to different viruses and different parasitoids during different parts of their life cycle. They're also responding to chemicals differently and to different stimuli. So these guys are really interested in their hosts, while these guys really don't necessarily care about hosts. Males, maybe, we'll see. That's work that a cat student is doing in the lab. And then we also have these differences in metabolic processes. So these guys are eating while these guys are using up any stored resources. So we did experiments row to look at these different Go categories. And this is essentially kind of like those black correlations plots that I showed you before. And as you can see, they're pretty highly coupled. Spearman's row is pretty high for both cellular localization and cell cycle. And there's not a big spread here between the different metamorphic events either. They're pretty on top of each other. When we look at the decoupled genes, we're seeing that there's not really a huge shift for the minor metamorphic stages, but that there's increased decoupling in pigmentation, immune system, response to stimulus, and lipid metabolic processes for those intermediate and complete metamorphosis events. But we all know that there's a problem here, right? Go terms, they're kind of tricky. So what we wanted to do was look at a set of manually curated genes as well. And we chose the chemosensory genes because of their role in the life cycle. And we looked at the ORs, which are the odorant receptors, the gustatory receptors, and the odorant binding proteins. And so we looked at 77 genes total in this species, and this is where we really see it, right? You have lots of a big change in decoupling, especially compared to those early stages in the cryptic and aposematic larvae, the aposematic and the wandering larvae, and the wandering larvae and the adults. And so we could guess which genes should be under antagonistic selection and therefore be decoupled. And so I hope I've convinced you of that. I'm going to let the next slide load. And it's going to show you that correlation plot again. So if you remember those correlation plots, there was a lot that are like kind of clustered here, but then there are some along these edges. And those are the ones that I'm interested in, right? So those are the genes that I predict are going to be evolving faster, right? So if we have this decoupling and the adaptive decoupling hypothesis is true, then those genes should be evolving faster because they're not under any of those antagonistic selection and pleiotropic constraints. And I think that they could play a role in adaptation. So if you've seen any of the other talks from the Linen lab today, you'll know that these guys don't just use one species of pine, they use a whole bunch. And host shifts are pretty common in this species. And so I wonder if we can use some of these genes to see if they pop up more when they're adapting to a new environment. And then finally, do they play a role in speciation? If they're involved in adaptation, especially to a novel host plant, do they also play a role in speciation? And I'd really like to thank all the members of the Linden Lab, especially Kim Vertaknik and Anna Soso, who started collecting some of this larval tissue before I joined the lab. And a second thanks to Kim Vertaknik because she not only curated the odorant receptors and gustatory receptors, she also taught me how to do the odorant binding proteins as well. And with that, I will take any questions from this very cute little soft light that is very confused with one male and one female antenna. So, email me if you have any questions.